All right, everybody, here we go. We are officially starting Unit 5, all about heredity. So the first part of that includes Chapter 10 in your book, which focuses on meiosis or meiosis. You could really say it either way. I think I'm going to unconsciously say it both ways, so just be prepared. And also the life cycles, whether it be sexual or asexual reproduction. So the first thing we need to cover are some vocabulary words. So genes are just segments or chunks of DNA that actually code for what will eventually become proteins, our basic units of heredity. And we know that our DNA is not all 100% genes, but we have about 30,000 genes inside of us. Offspring get these genes from their parents by inheriting chromosomes. So we have learned that humans have 23 pairs of chromosomes. And really, these are condensed versions of our DNA. And each one of those chromosomes has tons and tons of genes. There are two main types of reproduction. We as humans obviously do sexual reproduction, but asexual reproduction is done by bacteria and other like organisms. They essentially are cloning themselves. All of the offspring are genetically identical to the parent that they came from. So it's a single parent, and there's little variation because all of the offspring are exact clones. And there can be mutations that arrive randomly, and that can yield some variation between organisms, but generally speaking, they're all the same. It's fast because you only need one parent. You don't have to find another. It's energy efficient. Usually this happens by either budding off a little piece or something called binary fission, which basically just means it kind of cuts itself in half. Sexual reproduction, on the other hand, is meiosis, and that is where the organism produces gametes or sex cells. For us in humans, we have eggs and sperm. There has to be two parents, a male and a female, for sexual reproduction. And when you do this, it generates a lot of variation and diversity because depending on the male and the female, and depending on the specific combination of chromosomes that occurred in the egg and the sperm, you're going to get a lot of variation. It is slower. It is much more energy consuming, but because of that advantage of genetic diversity, a lot of organisms do this. So humans, plants like trees also do this. And here's an example on the left, a small microscopic organism called a hydra. It has a little bud hanging off the side that will eventually become its own organism, and then trees. We don't really focus on plant reproduction per se, but if you think about, pollen needing to go from one plant to another, they can be self-pollinating or to another plant. That's why we have bees. That's a form of sexual reproduction. Okay, so chromosomes. I mentioned some of this before, but you have, as a human, 23 pairs of chromosomes. 23 from mom, 23 from dad. So we have a total of 46 chromosomes. Every somatic cell, so we have somatic cells and we have gametes. Somatic cells are your body cells. It's pretty much everything in your whole body that is not an egg or a sperm. And so all of those cells have the entirety of your genetic material. They have 46 chromosomes, or you're going to see it written as 2N, meaning kind of two sets. We'll learn later that the phrase for that is called diploid, di for two. So you have two sets. Again, 23 from mom, 23 from dad. Each pair of homologous chromosomes, which is another vocab word, includes one chromosome from each parent. So what that means is you have two chromosome 1s. They are homologous, meaning that they're both chromosome 1, but one came from mom and one came from dad. You have two homologous chromosome 2s, two homologous chromosome 3s, and so on. So an autosome are the chromosomes or the pairs of chromosomes that do not determine sex. Again, these are the 22 pairs that are not your X and Y chromosomes. Your sex chromosomes, therefore, are that 23rd pair. And you will either have two X's if you are a female, or you have an X and a Y if you are a male. So that Y chromosome determines gender of the offspring. Gametes by themselves, the egg and the sperm cells, they don't have 46 chromosomes. They have only 23, which makes sense because when an egg and a sperm comes together, then it kind of leads to that double set. So that's why it's referred to as 1N instead of 2N. They have the 23 total chromosomes, 22 autosomes, and then one sex-determining chromosome. So an egg has the 22 from mom plus an X, and a sperm has 22 from dad plus an X, or a Y, which is again why the sperm determines the sex of the offspring. Okay. So again, here's another picture capturing roughly what a homologous chromosomes mean. And there's a lot of vocab here, so it's a little bit tricky. So let's say mom's chromosomes are in red and dad's chromosomes are in blue. In this case, there are a total of 6 chromosomes or 3 pairs. Because there are, let's say, 2 chromosome 1s, they are homologous. So we have dad's chromosome 1 in blue and mom's chromosome 1 in red. 2 here at the bottom, we have another pair of homologous chromosomes. And then the little guys would be a third pair of homologous chromosomes. So our total number, or both pairs together, have 6. 3 were contributed by mom, 3 were contributed by dad. Other vocabulary words here, this would be after the chromosomes have duplicated, so they have sister chromatids. So really this cell would have gone through S phase but has not divided yet because the chromosomes are in an X-like fashion, even though they don't really look like an X. The ones that are attached together are called sister chromatids, but together they are one chromosome. And they are joined together at the center by a centromere, which isn't really clearly pictured here. The actual picture representation of chromosomes is called a karyotype. And again, these are really done on cells and stained and laid out, and they try to then computer arrange them from largest to smallest. So your chromosome 1 is your largest chromosome, your chromosome 22 is your smallest chromosome, and then they always put the X and the Y at the end in the bottom right, and that will show you whether or not it's a male or a female. Again, it's roughly hard to see, but there's they usually take them in metaphase, but doesn't have to be. You can see that there's a pair of homologous chromosomes. So this would be one parent's, and this would be another parent's, and they are duplicated. So they are their own little tiny X's, and it's kind of hard to tell, but then you can see them splayed out over here. Here's another version of what that looks like in a little bit less colorful form. But they use karyotypes oftentimes to confirm or look at chromosomal disorders. You can't see individual genes on a chromosome, but you can generally see if there's an extra one or you're missing one. You can also determine sex. So for example, this one is a boy. There's an X chromosome and a Y. It is important to note that the Y chromosome is really small. So just by the fact that it's smaller does not necessarily mean that there's any kind of genetic condition. And again, you're kind of staining them and getting a picture of them. So if they don't look identical, that doesn't mean there's a genetic problem. It just means it's not laying nicely in the photo. But you can see, again, extras or missing ones. So in this case, there's an extra 21st chromosome. There's 3 instead of 2. So this particular individual would be a male born with Down syndrome. It's important to note, too, that cancer cells are also a little bit different. Oftentimes, because they divide without going through repair or apoptosis, they can have an abnormal number of chromosomes. And so this is a picture of a karyotype of a metastatic melanoma. So melanoma is skin cancer, the kind of black dots that you get. It's the most deadly form of skin cancer. But you can see this person has 3 chromosome 1s, 6 chromosome 2s, 3 chromosome 3s, 2 4s, 3 5s, 3 6s, and so on and so forth. So it's kind of all crazy. And again, there's not really the pressure to keep it regulated because they are cancer cells. Taking a side note, HeLa cells are the oldest and most commonly used cell line to study cancer. I worked with them once upon a time in graduate school, but they are a specific cell line taken from a person called Henrietta Lacks. And so that's where the name HeLa came from. He from Henrietta, La from Lacks. And they are really cultured cells from her cervical cancer. It's a really fascinating story. There's a book about it called The Immortal Life of Henrietta Lacks, because these cancer cells live on in scientific testing. But they actually took them without her knowledge and put them on a petri dish and really have been growing them ever since. And they have all kinds of crazy number of chromosomes because they've been dividing since, the early 50s, and they're still being used today. And the book is called The Immortal Life because cancer cells are basically immortal. They don't die when they normally would. And they have an active version of an enzyme called telomerase that helps cells live longer. And we'll learn about that in Unit 6 when we talk about DNA replication. But as I mentioned, they're pretty much the major cell line that people use to study cancer treatments. And they try to essentially kill them as a way to find cancer treatments. All right. So switching gears, we are now going to talk about life cycle. So in general, a life cycle is basically just the life and reproductive history of an organism, from when it is conceived all the way really till it makes offspring. And we know that obviously people live after they have offspring. I can be a case in point. But a life cycle generally is the generations that follow. So in terms of a sexual life cycle, it kind of alternates between 2 things: fertilization and meiosis. So initially in a parent, for example, meiosis occurs to generate gametes, the egg and sperm or sex cells. So there is a cell division that basically takes a cell that has 46 total chromosomes and divides it in half, so that way they go from diploid or 2N to haploid 1N. And then that creates one set of genetic material that will go into the next generation. And that is alternated with fertilization, where you actually combine an egg and a sperm together and create a fertilized egg that has gone up to now 2 sets of genetic material to 2N. And that is called a zygote. So it's called kind of an alternate because you go between having to create sex cells and then doubling genetic material upon fertilization. And then the zygote, once formed, divides by mitosis, which is what we learned before, where cells are kind of making copies of themselves. They're all diploid. They all have the total of 46 chromosomes. Okay, so the thing that I just described essentially is on the left, animals. So we have an organism like a mouse that is 2N or diploid. It has 46 total chromosomes. A mouse doesn't really have 46, but extrapolating 2 total sets, one from mom, one from dad. Then it's going to create sex cells that are halved. So these gametes would be egg or sperm, depending on the sex of the animal. And then one of those gametes will come together with a gamete from another organism. each containing half the genetic material. It will then be fertilized, and voila, we now have a zygote that is 2N again. So that's why it's considered an alteration of generations. And we have it color-coded so that pink is diploid and blue is haploid for ease. Now, plants roughly do the same thing, right? They have a zygote also. They have a diploid organism. It's got a fancy name of sporophyte, but you wouldn't need to know that. It also creates sex cells, in this case, spores. Those spores can actually reproduce on their own, but then they come together and fertilize and then can make a zygote. So plants are interesting because they have a haploid version that can survive, and they also have a diploid version that can survive. So it's kind of a little bit different, a little bit interesting, but they do have both. In this case, most fungi have a 2N version. They do go through meiosis and mitosis, and they can come together and fertilize. All of these are examples of sexual life cycles because they have different parents coming together to have a fertilization of some kind to generate offspring. Humans are pretty much the same way, as I mentioned. Females generate eggs, males generate sperm, and they come together. And then once that zygote is created, it will develop via mitosis. Okay, so now we're going to get into the process itself of meiosis. How do we go from having 46 chromosomes in a cell to having 23? And what does that look like? Well, it's a series of 2 divisions, and we're going to go through them. And at the end of it all, you're going to have 4 daughter cells, each with half as many chromosomes as the parent. And that could be egg cells or sperm cells. So normally in interphase, you have a pair of homologous chromosomes, right? You got your 23 pairs from mom, your 23 pairs from dad. And then during S phase, those all get duplicated. So that's kind of just a normal part of the cell cycle. And then voila, we have 46 chromosomes total with their copy. So that's a lot of genetic material. Now, if this was going through mitosis, then it would just go through IPMAT, prophase, metaphase, anaphase, telophase, and just divide, and you would end up with 2 daughter cells, the same as the parent. But that's not our goal here. In meiosis, what happens initially is that the sister chromatids actually stay together, and instead of all lining up vertically down the middle, the homologous pairs line up and then they separate. So you can get kind of a random combination. And then that's called meiosis 1. And at that point, you've essentially halved the genetic material because only half of the chromosomes came over here, even though they're with their copy, and only half came over here, even though they're with their copy. And then in meiosis 2, it's essentially mitosis. Those sister chromatids will then separate, all of them line up down the middle, and you end up with daughter cells. So we're going to kind of go through what that looks like. They still have the same name as in mitosis. There's prophase, metaphase, anaphase, and telophase. There is still cytokinesis because cells are dividing as well, but they're given 1 or 2. So meiosis 1 is the first division. As I mentioned, chromosomes are replicated. In prophase 1, there's something called synapsis, and this is where homologous chromosomes pair up. So you can see how here there's kind of 4 of them. It's called a tetrad because there are 4 chromatids. So 2 chromosomes, let's say we have dad's chromosome 1 and mom's chromosome 1 with their copies. So it's really 2 chromosomes. They are homologous. When they join together, they are called a tetrad, but synapsis is when all of the homologous chromosomes pair up. This provides a unique opportunity for something called crossing over. When they are stuck together like this, chunks of the chromosome can kind of swap places. And that's unique because then it can add to the diversity of offspring that can result. This picture shows what a chiasma is. It's really where that crossing event might take place. So there's centromeres, and there's an opportunity for some of them to kind of cross over. I think there's some slides later on about it. In metaphase 1, which is really what's pictured here, they all line up in the center, and then those homologous chromosomes will separate in anaphase. Sister chromatids are still attached together, so there's still an X happening for each one of those chromosomes. And then in telophase 1 in cytokinesis, you end up with half of the genetic material. Each chromosome is still stuck to its sister chromatid, and so on. Here's again what that might look like. And this is unique because it can show you in prophase 1 that cool, unique combination. So we have tetrads here, and when they are stuck together like that, you can see a chiasma where there is crossing over taking place. And then you can kind of see what that looks like in anaphase because you can see that a couple of chunks of swapped chromosomes have taken place. So then you end up with a little bit uniquer chromosomes at the end. All right. And then there is meiosis II. So this process is not quite done yet. You have the genetic material, but they're still attached to their sister chromatids. So in order to create the sex cells or gametes, it's essentially mitosis. So there's no interphase, so we don't double any DNA again. There's no crossing over taking place. Really, the spindle forms, the chromosomes line up, they separate, and each one of these cells will split, so you end up with a total of 4 at the end. And they're all a little bit unique because of that crossing over. So there's prophase, there's metaphase, here's anaphase, they're pulling apart, and then telophase, and then they're separating. So you can see here that each one again has slightly unique genetic material. So the eggs and the sperm are not exactly the same. So things that are unique to meiosis, meaning they do not happen in mitosis. There's crossing over events at the synapses or chiasmas. In metaphase, instead of just each chromosome lining up, it's really the homologous pairs lining up in their tetrads. And then in anaphase, when they pull apart, the sister chromatids are still attached together. So these are things that do not occur in mitosis that are unique to this meiosis I part. Okay, so I mentioned genetic variation and how crossing over helps to enhance that. And genetic variation is good in the grand scheme of things because the more genetic variation you have, the more likely a species will be able to survive whatever conditions it's in. So this crossing over allows you to recombine chromosomes and get unique combinations. What's also important as a piece of genetic variation is how the homologous pairs line up in metaphase 1. It's not to say, like, I guess there is a magical combination where all the paternal chromosomes could line up on one side and all the maternal chromosomes could line up on the other. And then when they split, you get this combination. But really, that's not how it works. Tetrads, when they line up, or each homologous pair, it's kind of random as to which one's on which side. So depending on how that all pans out, you're going to end up with very unique daughter cells. And that's called independent assortment. Their lining up is very random. And then on top of that, you get your, unique egg and your unique sperm, and they come together, and then those two sources of genetic material create a unique individual from there. Okay, so now we're going to combine both of the two together. Mitosis, again, is for your somatic cells, your body cells, whereas meiosis is for your sex cells, gametes. Mitosis has 1 division, meiosis has 2. Mitosis produces 2 diploid, meaning total genetic material daughter cells that are identical to the parent, which is why it says clones. Meiosis produces 4 haploid daughter cells that are genetically different. Mitosis is what your cells go through from when you're formed to when you die to grow and repair, and there's no crossing over. Meiosis is for the generation of eggs or sperm for reproduction. And then here's kind of the picture of those two things together. And then here's kind of a table at the very end that shows you how those go together also, or compares and contrasts. All right, you made it to the end.